My name is Thomas Schulze, and it's a pleasure to be with you today, albeit in a virtual format. So normally, if I were in beautiful Pakistan, I would ask you, "Kya hal hai? Aap kaise ho? How are you doing?" In Lahore, I would maybe try a few words in Punjabi, but today let's stick with a few words in Urdu. So my name is Thomas Schulze. I'm a professor at the University of Munich and a professor at SUNY Upstate Medical University in Syracuse, New York. And I am on the executive committee of the WPA. I'm the secretary for scientific sections. So there are several hats that I'm wearing. And when it comes to research, genetics of mental illness, of mental health is my topic. And today I want to talk to you about the genetics and pharmacogenetics of bipolar disorder. So let's advance the slides. When we talk about genetics in psychiatry, we basically talk about complex genetics research. That's not only something we do in psychiatry, that is in all kinds of well-known common disorders. These are complex disorders. And what are the strategies that we use to unravel the genetics of these disorders? So we use various strategies, and we have been using them for quite a while. I may say for 100 years. So everything started with family twin and adoption studies that have laid the foundation for us knowing that there is a genetic basis to mental illness. That was followed by genetic linkage studies where researchers collected large pedigrees of patients and their relatives and large families with multiple affected individuals. And they would try to find through genetics, through statistical means, regions of chromosomes that would presumably harbor genetic risk loci, genetic vulnerability loci, or susceptibility loci, as you may call them. Another study were candidate gene studies, whereby you would test a hypothesis that a certain gene, or a gene variant actually to be more precise, is more present in cases than controls. You could do this with cases of controls, but you could also do it within families with parents and offspring. This is the so-called parent-offspring trio approach. Well, this was the situation until the year 2000. So that was what dominated until the year 2000. These kinds of studies, whereby, of course, family twin and adoption studies were done in the mid 20th century, maybe in the 50s and the 60s and 70s mostly. But still, these were the traditional approaches that, when I started in the field in 1997, were the ones that were around, that were available. But the year 2000, something happened. The whole genome was presented to the world. There were initiatives, the Human Genome Project. There was a private initiative, private enterprise, and a public enterprise. And the results of that were presented in Nature and Science. And what was presented? Basically, the whole length of the genome. So you now knew which base pair is at what position of the DNA. That was an amazing and a major feat, because now we were in a position to do studies in a much denser way. Before, we would use a few markers that were spaced at large distances across the genome. Now we were enabled to study many, many more polymorphisms. In fact, this is now the basis for all what we do today, because now we are using genome-wide association studies. We use the whole genome. We use markers that are spaced at a short distance and compare cases and controls. beyond that, or I would say we have already reached beyond that, because now we're not only relying on GWAS, we are also trying to explore further. Because a GWAS is basically a study where you buy a chip from a company that has several hundreds of thousands of markers, or even or million of markers on there, and you test them. It's like going to a department store and buy something, right? But it's not. It's not custom made. It's not tailored. So, but it is conceivable that there are, of course, variants that may only be present in certain populations, in certain families, or even individuals. And these you can find through sequencing, by really unraveling each and every, or turning each and every stone, every brick, every base pair in the DNA. And that's what we're doing as well nowadays. So I said it's a little bit of a journey. I think it started, one could say, the early 20th century, and we are now in the early 21st century. So it's pretty much 100 years. When we do genome-wide association studies, we do not anymore genetics, looking at a few variables or few variants, I might say, but we are looking at the whole genome, right? The O is prevalent. It's genomics. You know, when you look at the whole proteomics, the whole proteome, you do proteomics. When you look at all transcripts, you do transcriptomics. Then there's epigenomics. And we also talk about phenomics, when we try to capture the whole phenomic space. So we're doing genomics. We have now big data, large samples, large cohorts, and novel and powerful molecular tools at our hands. Hence, we can perform case control studies in thousands and tens of thousands of individuals using these modern technologies, aka here, genome-wide association studies. Now, you may be familiar with the significance threshold, right? We typically use p-value of 0.05, type 1 error of 0.05. That's the error probability that we accept. That's a p-value, and if it's below, if we test the hypothesis and the p-value is less than 0.05, we are happy, because then we claim significance. However, when we deal with genome-wide association studies, we are testing so many hypotheses, it's in the millions, we have to adhere by more stringent standards. That's why in genome-wide association studies, we're using a significance threshold that is much stronger. I mean, the p-value is much smaller. It's below 5 times 10 to the minus 8. This is the p-value that you have to surpass or have to reach to claim significance for a tested marker. Now, as I said, genome-wide association studies are not something that are unique to psychiatry. Every field of medicine, every complex genetics analysis or complex medicine field uses that. Now, this is a cartoon from the GWAS catalog that was compiled at the NIH or put together at NIH or is maintained at the NIH. That is a screenshot or download from 2010. There you see all the chromosomes from chromosome 1 to chromosome Y to chromosome X. And you see color-coded traits or phenotypes with a genome-wide significant finding, right? It's kind of crowded, right? So, and when you look closer, you will see that you have a lot of traits and phenotypes. And it's not only diseases. It is like, well, you have like longevity, liver enzymes, malaria, glioma, bilirubin, autism. So that means back then we had 1,212 published GWAS studies, with at least one genome-wide significant finding. Now you can imagine that the field having advanced, we are now at a more crowded picture, right? And there we go. This is now the GWAS catalog screenshot from What is that? 2013, just 2 years later, 3 years later. Now you see it's pretty busy, and actually it's not anymore single traits or phenotypes, it's trait categories, because otherwise you wouldn't be able to plot that anymore. Trait categories meaning 17 larger systems like digestive system, cardiovascular, metabolic, response to drug, cancer, and so forth. Moving forward even further, or fast forward, even not fast forward, but going into basically real time almost, you will see that this is even busier. This is now from 2019. Still 17 categories, and now look at that. You can hardly discern any single dot anymore. That means we have so many genome-wide significant findings for so many traits, for so many physiological traits or disease phenotypes. I'm just showing you that to tell you that what we do in psychiatry is what we do in other fields of medicine. It is standard analysis, and I might say that this is like a tool now. It's a workhorse, genome-wide association study. This is something that has become so abundant and so routinely used that it is, an everyday workhorse in our field. Now, when you want to do that, when you want to do such study, you cannot rely anymore on small samples, right? You cannot test a genome-wide association, or you cannot look for genome-wide association in a small sample of 100 controls and 100 cases. That's not going to fly. There's no power, right? Given all the tests you do. So you need to work in large groups, in large consortium, which we do. We have, for instance, the Psychiatric Genomics Consortium. It's a great endeavor. Groups from all over the world join forces by joining brain power, by joining resources, computers, computing facilities, by joining samples, by joining data, by joining, by sharing, or by sharing in writing papers, publications, creating new resources, sub-resources. It's an amazing feat. This is really how science should work, and actually that's the way science does work now, is particularly now in psychiatry. So this is a consortium that each and everyone who is interested may consider joining. And with that power of sample size, with that power of logistics, with that power of manpower, with that power of spirit that's represented there, you can achieve amazing things. So the first study that came out was on schizophrenia, and we were able, given the large sample sizes, to identify more than 100 loci for schizophrenia. Now the field has advanced, and we have now more findings. So just to give you a quick sneak preview or so, that's about to be published. It's already in a preview, in a pre-publication. This is the result of the latest schizophrenia GWAS. The latest schizophrenia GWAS from the PGC, from the Psychiatric Genomics Consortium. And we studied almost 70,000 cases, more than 230,000 controls. And we identified 270 distinct loci, meaning 270 loci markers, single nucleotide polymorphisms, or SNPs markers, that surpass the threshold of genome-wide significance. Now, just to interpret that graph here, which is known as a Manhattan plot, because it's thought to resemble the skyline of Manhattan, you have the y-axis, which is the -10 log 10 of the p-value, and then you have the chromosomes. Now, you also have a line that can hardly be seen anymore, given the crowded nature of that plot here, but this is the threshold of genome-wide significance. That is the p-value threshold of below 5 × 10⁻⁸. Every marker, every single nucleotide polymorphism, every SNP that is above that line is one of those 270 loci. They are associated at a genome-wide significant level with the disorder in question. Now, you see that some show an amazingly small p-value, which doesn't always say anything about the strength of the association, that would be the odds ratio. It just shows you that this is really very good significance, right? And it's not just borderline significant. And this is something that will most likely hold up in subsequent studies. In fact, it is a region in the major histocompatibility complex, underscoring the immune hypothesis, or the immunological hypothesis of schizophrenia. So that is how these results are presented. Now, it is our job, the research community's job, to do something with this, right? We can go now, zoom in, what do all these markers do? We can test them in models, in cellular models. We can test them in animal models. We can maybe design drug trials based on the knowledge we gain from that. So there's a lot to do with these results. Or we can use them as a composite score. We can score each and every individual for the presence of these risk variants. And then this is called the polygenic risk profile, or risk score. And every person can get such a score, and it basically will then tell, this variable will tell the genetic loading of a person for a certain trait. Now, similar things are being done for depression. We are not that advanced yet in terms of sample size. We are now up to more than 40 variants for depression. Sample sizes are going up, but at that point, we are not there. We are not where we are for schizophrenia, and depression being quite heterogeneous may be a harder case. For bipolar disorder, this is the state of the art a few years ago. We had identified 30 loci, but things have evolved, and I will show you the latest results in a few minutes. So where do we stand in psychiatric genetics? Well, when I started in 1997, I was told to recruit large families and do a few analyses, and I would most certainly be part of the final discovery of risk genes for a given disorder, because the idea was that there would be a handful or 10, 20, 30 genes, major genes, that is an oligogenic model responsible for schizophrenia or depression or bipolar disorder. Now we know that is not the case anymore. We are talking polygenicity here. We know it is not anymore a few individual genes. It will be hundreds, if not thousands, that interact, each of these genes only contributing a tiny wee bit to the overall genetic risk or liability. But I think we still have made progress, even tremendous progress, because we have identified genes and we have the evidence that I think it's a game changer to know that this is a polygenic disorder, or these disorders are polygenic traits. We also have a clear evidence, and I'm not going into details here, of other variants that are not represented in the GWAS, or we have a clear evidence of rare genetic variations, such as C and D's, copy number variants, intellectual disability, autism, and partly schizophrenia. And these variants that are rare, but confer a higher risk, are now included even in recommended genetic testing strategies for workup for diagnostic workups in intellectual disability diagnosis making or autism and so forth. And the discussion is out whether we should also consider something like that in early psychosis cases. So clear evidence of polygenicity, I think, is a major, major achievement. It's a game changer. And although it's maybe not what we wanted, right? These 10 or 20 genes that explain everything, We have learned, maybe the hard way, that it's polygenic. We have to live with it, but that's okay. We now know how the architecture in theory is. We haven't really sorted it out in detail how it works, but we know it's polygenic. And we may have novel leads that warrant further analyses. So consortia efforts have been key. Since the statistical power comes with increasing sample sizes, I think we have known that all along, have we? So, but maybe not. Now we know it. Some disorders are more heavily researched than others, and this is not a function of their prevalence. It's interesting. There are more schizophrenia researchers than there are major depression researchers, although depression is much more prevalent. It's just an observation on the side. Increasing sample sizes alone, however, will not be the silver bullet for a deeper understanding of the underlying genetics. I would argue that we need to explore the vast phenomic and multi-omic space. What does it mean? Look at the phenotype, not just the diagnostic phenotype, DSM-based or ICD-11-based, whatever phenotype. Look at more in-depth clinical phenotypes that are relevant to clinical psychiatry, and do multi-omic approaches. That means look at the transcriptome, at the proteome, at the methylome, that is epigenome, maybe even microbiome nowadays, right? There's so many ohms we can look at, and we should do it. Let's dig deeper a bit into bipolar disorder, because that's what the talk mainly is about. This is the latest GWAS, published a couple of months ago. Almost 42,000 cases, more than 370,000 controls. You see, it's a consortium. You can hardly read the authors anymore, which is great because it shows that this is a consortium endeavor. We have 64 associated genomic loci. And what is nice is this study confirms many, many previously associated loci. It also detects novel loci. We see, for instance, bipolar risk alleles that are enriched in genes in synaptic and calcium signaling pathways and brain-expressed genes like prefrontal cortex, hippocampus. We see a signal enrichment in genes encoding targets of antipsychotics, calcium channel blockers, anti-epileptics. We even have identified druggable genes such as these guys. I'm not going to pronounce their names now. You may want to look them up. It just shows you that there is a huge potential in there. And the larger the sample size, the more we can consolidate findings, especially when we confirm previously associated loci and when we don't confirm them anymore. And we get this huge bag, or treasure trove, right? Treasure chest of information that we now need to dig further into. When I say we need to do more phenomics, I would, for instance, recommend doing or looking at the age of onset and polarity of onset in bipolar disorder. Postdoc in my team, Janos Kalman, has been looking at that. He looked at all the samples of the PGC, bipolar, and asked the investigators to provide him with information on age of onset and polarity of onset, which actually was we thought might be easy, but actually was not. It was really, really tiring and tedious. It took us 5 years of work. You've seen we had more than 40,000 individuals, but we don't have 40,000 individuals with this detailed phenotypic information. We unfortunately, but it's in a way unfortunate, we only have like 13,000 cases. We said of 13,000 individuals with that information. We set aside roughly 2,000 that are meant for replication purposes. This is for age at onset, and the numbers for polarity of onset, whether you start with mania or depression, are much lower. But that's how it is. That's the reality. Why is that? Because it's hard to get all that information, right? Some people have a limited recruitment program. They can only record out of their clinical records the diagnosis, and they may have time to get some blood or some swab to get saliva, and that's it. So, but ideally, we should have more information, detailed phenotypic information, at least for phenotypes that are so important in genetics, like age at onset. But we have to live with what we have. But I think I would also make this an ad. I would like to advertise, I would like to advocate for studies that not only recruit cases based on the diagnosis, but also record detailed phenotypic information. The information that we obtained was very heterogeneous. Why is that? Because age at onset is defined in different ways. But we have to live with that. We just have to record it. In the end, we didn't see anything significant for polarity of onset, but we saw some significance for age at onset. We have a gene that is actually implicated in obesity, FGO. It's a well-known gene. We found that, and which is nice. We unfortunately could not replicate that in the smaller replication sample. Well, we could have guessed that, right? I mean, you find something in 12,000 and then you want to replicate it in 2,000. We wanted to have a stringent approach, meaning we wanted to have a replication sample at hand. We set that aside. Had it been maybe a stronger finding, it may have replicated in that sample. We don't know. So it doesn't replicate. But I think it's a first lead that warrants further analyses and actually calls for more analyses of that, for larger sample sizes, for going back to the phenotype, for digging deeper into the phenotype, for adding more phenotypic component, even retrospectively to sample collections. And when we use the profiling score system that I showed you, this polygenic profiling scoring, we can, for instance, demonstrate that higher genetic liability for autism spectrum disorder, major depression, and schizophrenia is associated with an early age at onset. What does it mean? You have a polygenic score, a polygenic profile with a higher load for schizophrenia risk alleles. You're not schizophrenic, but we score your genomic information based on the information we have from schizophrenia data, and we can see that the higher the score is for you, the more likely you have an early age at onset for bipolar disorder. And interestingly enough, we also see something about educational attainment, but this is something that we also sometimes see in the clinical observations. You will find more highly educated people in bipolars, right? So a higher educational attainment is associated with an early age at onset. So there are some connections, or there are some relationships, hidden relationships, interesting relationships that need to be fully understood, that you can try to unravel with this method. So we can say that age at onset of bipolar disorder is influenced by a broad liability for psychiatric illness. But we need larger and better characterized samples, and the clinical input is always key. Now, when I say clinical input, I would like to move a little bit beyond only including age at onset, for instance, or psychosis. I would like to do something, and I've been promoting that. I would like to go longitudinal. So we in Germany created a multi-site resource with centers in Germany and one center in Austria, where we recruited in a longitudinal fashion schizophrenia, bipolar, major depression individuals and controls. We have roughly 1,700 individuals. We have interviews on them at four time points, 0, 6, 12, 18 months, and we have complete biobanking at each time point. This allows us to study the genetics, the genomics, the multi-omics of the longitudinal course of psychosis. And we are about to use that resource. It took us 10 years to complete that. Only now are we starting to use that and to run analyses with various aspects. This resource is available. It's called psycourse.de or Psycourse, and it's that website. This is the paper that accompanies it. Just go there and you see it's open for open science. You can apply for the data, and we would then see how we can accommodate your requests. And many requests have already been made, and they have basically all been granted. And so this is an open science scenario. And I invite you to take a closer look. We have a detailed code book on the variables that we have and so forth. Another phenotype that is very dear to my heart is lithium response. We know that lithium is a drug that actually works, well, maybe in 30% only, but we have a clear picture. When it works, it really works. So in 2008, we gathered at the NIH a group of researchers from all over the world that do research in bipolar disorder genetics and also are also interested in lithium. So we created a consortium called the Consortium of Lithium Genetics. At that time, the sample or the groups was just a few countries. And we have a website, www.condigen.org. There you can look that up. We put together all these samples that we had, bipolar samples with information on lithium response. We used a scale developed by Paul Groff and Martin Alda to assess in a lifetime fashion response to lithium. We described the phenotypic procedure here in that paper. And this helped us create the largest GWAS to date of people phenotyped for lithium response. And that's 2,500 individuals. And the GWAS showed, now you should know what that is. That is a Manhattan plot. It's not so Manhattan-like. It's not so impressive because it's a small sample, and we know sample size is key, but the phenotype may be quite homogeneous, which is maybe why we see significant results surpassing that threshold. When you zoom in, you see that these results are actually driven by these variants here, and they are more like proxies for each other. They're highly correlated. So these two are highly correlated, these two are highly correlated, but even these two clusters are highly correlated. So you may want to consider that one locus. And what these loci code for are not easily explainable genes. These are long non-coding RNA. That means RNA that are used or that play a role in genomic regulation. We don't know yet what they do. But it's interesting that we found that because this is a hot topic nowadays, non-coding RNA. We're going more into the regulatory realm of genomics. And there was a sort of a confirmation or corroboration. We looked at a sample from the VA hospital in San Diego, where a sample of like 70 people was studied in a trial where they were put on lithium monotherapy, and they looked at time to relapse. So when did they relapse? So you have it's an observation period of up to 120 weeks. So that's 2 years. And you can see the following. The green line is the line represents those folks that are homozygous, that have 2 copies of the response allele, the good response allele. And this is a line that represents individuals that have only 1 response allele. So you can see that the ones with 2 response allele show a longer time to relapse. So maybe this means if you have that genetic makeup, then you are more protected, then you have a better response. This was published in The Lancet, and The Lancet, this article inspired many folks all around the world because they see that we take authorship seriously, and they're all authors, 124 authors are on the paper, and at that time we convinced The Lancet to do that, which was unheard of in these days. And that, I think, inspires a lot of folks all around the world. We held trainings in Peru, in Ethiopia, in Bolivia, in Sudan, in Ukraine, in Azerbaijan, and we're happy to hold meetings maybe at Fountain House in Lahore at one point. And hopefully by doing this, getting more groups involved and increasing the sample size. That's what we're actually trying to achieve now, bumping up the sample size for Condigen. So Condigen is trying to recruit more groups all over the world. And you see the yellow sites. These are the sites that have expressed interest. That means the consortium has grown. This is now our group of countries that is represented. I would like to spend a few words on pharmacogenetic testing. You may be familiar that these tests are already available, and companies say, well, you can save a lot of trial and error approaches. You can increase compliance by doing such a testing a patient, and the patient will then you will have a result for the patient's genetic makeup, and you know which drug to take and which not, which drug will work, which drug will not work, and so forth. There's some rationale behind it. The question that I would like to highlight is it ready for prime time yet? Are these tests that are sold, sometimes direct to consumer, are we in a position to use them routinely? That is, before you do anything, before you take a history, before you interview the family member, before you look at prior information, and before you order medical records from another hospital, should you run a genetic test, and then say, hey, the test says you should have venlafaxine because for you, citalopram will not work. And should you then do this based on a test? This means, is a routine use of a pharmacogenetic test something that we should endorse? The ISPG, the International Society of Psychiatric Genetics, whose president I was until last year, has come up with a statement, and we're still working on refining that. But we basically say, if you have such information from other genomic tests or so, you may want to use it. But this idea of routinely testing for pharmacogenetics is not that, that information that we get from these tests is not that robust yet. And some of the tests that are out there are really, honestly, reckless, because they are based on on studies that are based on a few individuals, because everybody can come up with such a test. There are tests that definitely adhere to higher standards. But the question is, should we already employ them in a routine basis? And should we basically sacrifice some clinical information that we otherwise would gather? That's a question, because most of these studies these tests, or most studies that are out there that show maybe the usefulness of the test were not highly powered. They were small sample sizes. We are now in a European consortium I helped put together, where we study various sites in Europe and America and Israel, more than 2,000 patients. We randomize them to treatment as usual and to treatment based on a genotype test. I want to see whether the routine application of pharmacogenetics actually will improve outcome. So what's my action plan that I would like to share with you? I think while we did not get what we initially hoped for, psychiatric genetics has made major progress. Collaboration has been key, so we should continue with that. We should broaden it, we should widen it, we should deepen it. But these collaborations, these efforts have so far been widely confined to cross-sectionally established samples with categorical phenotypes. We need to go longitudinal. We need to go more into the phenotypic variability. Let's add the input from the clinical field. Let's look at recovery, outcome, and what have you. You name it. Let's use this model and go longitudinal and dynamic to get a more complete and clinically useful biomarker framework. With dynamic, I mean, let's look at transcriptomics, because that's when a gene gets expressed. You know, a gene having a genetic variant doesn't say much. You want to know whether it's expressed, right? Whether there's a path from gene to protein that is active. So that's why you may want to also look into proteomics. What about epigenetics, methylomics? Well, these things cost money, but we need to do them. So pharmacogenetics has a huge potential, but it needs further vetting. And do not forget the patients and study participants' perspective. So in all that, the wider, the broader, the deeper, the longer, whatever, the more intense we will be performing that kind of research, the more we need to have the patient on our side. We need the patient's involvement. We need to ask the patients what they think about it, how we could improve the research, how we can make this research benefit them, more directly, not so much indirectly, because we know it's basic research. It's not really, most of it's basic research. It's not yet ready for clinical prime time. So with that, I would like to finish. I would like to say thank you very much, or shukriya. I would like to say Khulda Hafiz to the audience in Pakistan. I was there a long, long time ago in 1990, coming from East Turkestan along the Silk Road and entering into the Karakoram Highway into beautiful Hunza Valley and Gilgit, Chilas. Beautiful area. I didn't make it to Lahore, so I hope to be there next year. Been to Taxila, Islamabad, and so forth. Beautiful country you have. You're doing a great job there, and my dear friend Afzal and his team are doing a great job at Fountain House. So I am looking forward to implementing maybe our kind of research there at one point. And if there are questions, just Google my name. You find me everywhere on the internet. I mean, my internet, my email address, you can find me, send me a question, and I'm happy to respond. So again, shukriya, thank you very much, and Khulda Hafiz in Lahore, maybe next year.